Cool. So now we're talking about tissues. We talked about the cells. All tissues are different types of cells that are holding hands. 200 different types of cells. It just takes a combination of any of those to do it. But they have to have one common job. So the reasons that cells replicate to make tissues are for two reasons. Either the initial development to make the organism. So from the time that you were a sperm and an egg that merged together, that replicated and replicated and replicated. It became arms and limbs and organs and all that good stuff and skin, all from the exact same cell, all the exact same DNA, until you became the whole organism. The other reason is when you damage a part of your body, then that area has to regenerate, so then the cells have to replicate again. Like skin. If you have damage to the skin on both sides of it, here's a row of skin cells, they start replicating to fill in that empty area. So this cell will replicate and become 2, and that one becomes 2, and then 2, until you finally have that entire new covering. And then they're done replicating. Well, not completely, because they'll keep replicating and sloughing off the surface skin, but they quit moving inwards. Once they've patched or repaired whatever that is with new tissue, they're done. The process for doing that is called mitosis. So we'll talk briefly about the cell life cycle. You learned this in biology. Did we actually talk about it a little bit last week? Mitosis?
No, mitosis.
Perfect. So the life cycle. I got a lot of slides for it, but I can make it a little bit easier. For most of the cell's life, if this were like a clock, it sits right here where nothing's happening. Call that G0. There's no growth, there's no change, there's no nothing. The cell's just there, it's living, and it's doing its normal day-to-day business. But when a cell needs to replicate, or I like to think of it as having a baby, it starts going into a brand new phase. And this new phase takes up most of this circular clock. And it's called interphase. So like I said, I like to think of it like when a woman's having a baby. At the beginning of the first phase, which is called G1, or growth phase 1, this is where most of the gathering of nutrients, the building the structures, the collecting like amino acids and lipids and carbohydrates, just like when a woman gets pregnant, she starts eating more food to start taking in these extra nutrients to start building this new organism. It's the same idea with the cell. It's gathering all the materials, the raw materials, to start building. At the very end of that phase, there's a little checkpoint, and the cell evaluates, do I have all the raw materials I need? Am I ready to do this? Then if it is, it moves into the next phase, called the S phase. S for synthesis. Because in this stage, it synthesizes DNA. It replicates all the DNA inside that cell. And this is really important because in mitosis, M-I-T-O-S-I-S, it makes a perfect twin, which means all the DNA inside this next cell is going to be what? Exactly like or exactly different? It's exactly the same. It makes a perfect twin. When this process is completely done, basically it's cloned itself. You know what the other type? It's not meiosis, but there's another one that's like it. Yep. And I stress the I, so I always say meiosis, because same thing. Instead of mitosis with a T, it's the I for individuals. Oops. During meiosis, you make 2 brand new cells that are completely different than each other. The only time you use meiosis is when you make something called a gamete, which is either the sperm or the egg. So you pass on the shuffled or random assortment of genes down to the next generation. So what we're talking about now is mitosis. It's replicating a cell to make a perfect twin. And the third phase of interphase, or third section of interphase, is called G2. It's the second growth phase. You've got all the raw materials, you've got the DNA synthesized. Now all you're doing is arranging everything the way you want it, pretty much. And that's all interphase. You can see most of the time when a cell is replicating, it spends most of its life in interphase. Just like this. I guess I could have flipped that slide a little bit earlier. But here you can see that growth, the G1. There's the checkpoint. If everything's okay, then it proceeds through. It goes to the synthesis phase. And then it goes to G2, the final preparations and starting to build things. When it gets here, there's one more checkpoint. And if everything's not perfect at this point, that cell will try and fix whatever's wrong, or if it can't fix it, it'll commit suicide. So it goes through something called apoptosis, where if everything's perfect, it thinks, I can't replicate, there's something wrong with me, I'm just going to die. Otherwise, if it slides through this and it replicates with flaws, you could potentially have a what? A tumor. Yep, a tumor or a cancer. So it's a good thing that the cell does this checkpoint. Cancers are because these checkpoints break, and the cell can't stop replicating. So it's flawed, it makes all these mistakes, and then it just replicates and replicates and replicates. But that's interphase. And then at the very end of this, think of it as birth. As soon as you hit the second checkpoint, the G2 checkpoint, now it's time to give birth to this brand new cell. You've got all the DNA, you have all the structures you need, now you're ready to pinch this thing off. And then, oops, we already talked about that. And that's this part, mitosis. So there's several phases of mitosis. And all you need to know, I'm not going to, you learned it in biology, so I'm not going to repeat the whole thing in your head, but I'm going to point out the main parts. So the first thing that happens, while this cell is in interphase, the cell has a nice dark nucleus, the DNA is all replicated, it's all dark inside there, and it looks like a solid nucleus. As soon as you're ready to go into mitosis, things change. When you go into prophase, the first significant change. And here are the things you'll want to watch for. That nucleus, the nucleus dissolves. The membrane starts falling apart. You don't see a clear membrane anymore. I think I have a cartoon of it. I guess I don't have a really good cartoon of it. That's about the best. So down here you can see the nucleus starting to break apart until the very end they're just fragments of it. Second, chromosomes condense. Back here in interphase, all this stuff, all this dark material in here is just called chromatin. It's just DNA, but this collection of DNA is called chromatin. During prophase, it starts condensing and turning into actual chromosomes. So you can see all these wormy looking chromosomes all over the place. And the last one, these little glowy structures are called centrioles. And the centrioles move to poles. I'm putting poles in quotes. Because if you look at this kind of like the globe, now you've got all these little chromosomes in here. Up at the top you have these centrioles. I'm just drawing kind of extra represent them because they're little bars. They cross over each other just like this. And what they're actually going to be are puppet masters. I'll show you what I mean in a second. So even though in this picture it says early prophase and late prophase, I really don't care about the difference between those for this class. Just collectively call them prophase and know those three major things. Now that you've broken apart the nucleus, the chromosomes have condensed, now you're going to go into the next phase, which is called metaphase. The centrioles are still at the poles, the nucleus is gone, and all the chromosomes line up right along the center, or the equator, if you think of this like a globe again. And they call this the plate, the metaphase plate specifically. So you've got this metaphase plate right along the center. These centrioles are all connecting down to the chromosomes. And the bottom one is connecting up to the chromosomes. So the centriole, each pole, the north and the south, both of them have a hold of the same sets of chromosomes. Right now they're connected. So you form the metaphase plate. And don't think of this as just a line, it's actually a plate, because if you're looking at it like this, remember it's three-dimensional. If you tilt it to the side, you see they're all scattered across this plate-like structure instead of just a line. It looks kind of like that. So you can see the North Pole, the South Pole, and then right along the equator. It looks crazy. And then here you can see there's a centriole, there are all the strands coming down, microtubules are actually reaching down and grabbing hold. You've got your chromosomes lined across the center. It's not perfectly like the cartoon, but you can see they're all kind of lined up. And the next phase is they rip. So the next one, anaphase. Chromosomes split and move to opposite poles. Chromosomes split, move to opposite poles. So you see centriole, centriole. Chromosomes along this side, they're being pulled to the North Pole. You have chromosomes along this side, they're being pulled to the South Pole. You're ripped apart. That's the distinguishing characteristic of anaphase. When they rip and they go to opposite sides of the cell. Sorry, shoes untied. And the last one, telophase. Not a circle anymore, it's more like a peanut. And with telophase, the characteristics are that two new nuclei form. See it? So, two new nuclei form. And you can see this special structure called a cleavage furrow. Two new nuclei in the cleavage furrow. F-U-R-R-O, like right here. And then some books say cytokinesis is part of telophase, some books say it's separate. Cytokinesis is just a splitting. So the last step is that cytokinesis is that you have two brand new cells that are what? Total individuals or total twins? Yeah, they're perfect twins. That's mitosis. And now both of those cells are back at G0. They're back at the beginning. They're just living their life, individual cells. So you can watch the process on high speed there. Bink. They're all listed up here for you again. And then there's that process. Centrioles go to the poles, the metaphase plate, anaphase rips them apart, telophase starts cleavage furrowing, and then cytokinesis totally separates them. I'm going to put a couple slides going into practice. Like, if you're looking at this, what would that be? Yep, how about this one? They're all bunched up right in the center. This would be the metaphase. Yep. How about this one? Yep. And then how about this one, where they're now the two separate cells? Cytokinesis. So not so tough. Kind of a review. Actually, completely a review. Right, so they all have the same DNA. You became a zygote. You were a sperm and an egg. You merged together. Now you have half of your chromosomes from mom, half your chromosomes from dad. Now you have to grow. As that cell's growing, all the cells look pretty much the same, and some trigger flips on that says, now it's time to start becoming individuals. You're going to start looking totally unique. And what happens is they take the same DNA and start turning on different genes to become different structures. Like, for instance, it becomes a blastocyst, where it's a hollow ball with actually a little wad of cells in the middle of it. There are some things they know, but they're not completely sure why those things turn on. So, what flips them on? Pretty much. But why? We don't know. It's the same thing with puberty. When you start puberty, estrogen and testosterone turn on bone growth and make your bones grow really fast. At the end of puberty, guess what two hormones shut off puberty? The same two hormones. We don't know exactly why. We just know that they're both responsible. There's some kind of a clock mechanism that's built into our DNA that does it. We just don't know why. But then, huh? Are you taller than your parents? About the same height as your parents?
I'm about the same height as my mom.
So genetics.
I'm the tallest one of my sisters, so.
I'm about 4 inches taller than my dad and about 5 or 6 inches taller than my mom. I'm wondering how tall the mailman is. I'm recording that. I shouldn't say things like that. But anyway, then these cells start flipping switches on where they all have the same DNA, but they become different structures. Oops, that's right in the way. So you see, with the same DNA, you can become skin cells, you can become neurons, you can become a red blood cell, you can do all these different things with exactly the same DNA. It just depends on the triggers. And we'll actually talk about some of the triggers. And there's your egg and your sperm. They merge together, form a zygote. The zygote starts growing just a little bit, and then it splits the cell in half. So now you have two cells. Those two split, so you have four. those 4 split, you have 8, and then 16, and then 32, 64, 128, and just keep going like that until you have this big mass of cells. They'll start becoming very individualized, called differentiation, until you create all of you. And we'll talk about this at the end of the next semester too. This is embryology. But you start from one cell and you just keep growing and developing. You grow basically what looks like a tail, and then your legs and arms grow into it. Your head, you see the eye spot before it's an actual eyeball. You just keep growing. We all look like chickens or lizards or whatever.
First a tadpole, then a little baby T-Rex.
T-Rex. And here are some of the examples that we'll talk about. And you can see over 200 different types of cells, exact same DNA, but they all have different features. Like the sperm, it's the only cell in the whole body that has something called a flagella, a tail like that. In fact, haha, women don't get it, just guys. So we have a new unique feature that you guys don't have. Haha. And then smooth muscle. My girlfriend would say, you say, oh, talk like that, you'll never get any of those things. Right? So anyway, smooth muscles have special structures, all muscles, so that they can contract well. Nerve cells, you see the cell body, but they have long projections that come out so they can send a signal a long distance. In fact, the longest cell in your body is a nerve cell. It's a neuron that goes from the tip of your tail all the way down to your toe, which is roughly about 3 feet for most people. Epithelial cells, you can see how they have basically a jigsaw puzzle type of design so that they can stick together and hold hands. Or even the red blood cell that has that bent shape or weird shape because its ability to diffuse particles is a lot better by having that shape. So depending on what your need is, the right DNA falls into place and gives you that structure. Same thing for cells.
So like elderly, how their skin is easily more torn or cut?
It's thin.
Is that because the cells are thin, or is it because like the jigsaw puzzle's not so much a puzzle anymore?
Yes. Both. Yeah. Yeah, they have thin skin because the genetics aren't making the cells as fast. It's thinner, and then the connective tissues like collagen doesn't hold the way it used to. That's why you can also take and pull their skin up and it stays held up for longer. If you get dehydrated, you can do that too. My grandmother, she used to always play with her neck and do that stuff with her neck. She was well hydrated, but her skin was so loose there. It was gross. It was scary. So cell differentiation is the term for them turning into different things. And cell differentiation determines the shape of the cell and the amount of the organelles. Almost all the cells, with the red blood cell being an exclusion, all the cells have the same DNA and they have the same organelles. It's just numbers. So how many of each organelle? Like a neuron needs to make a lot of energy, or a muscle cell needs to make a lot of energy. So what organelle are they going to have a lot of? The mitochondria. They're going to have a ton. But when you look at a skin cell that doesn't need to make that much energy, it's barely going to have any mitochondria in it. So they're exact opposites. Where neurons, they don't have to grow and get bigger, so they have very few structures inside of them to make long protein structures. But when you look at something like a fibroblast that makes collagen, it has all kinds of rough endoplasmic reticulum to make these long protein fibers. So they both have the same structures, it's just how much of each do they need. And here's some of the examples that we'll look at, like I just said, fibroblast. And start getting used to the names. Blast means maker. What do you think fibro is referring to? Fibers, yep. So they're kind of like little spiders in your body. They start making these webbing structures all over the place, and they're designed to be really efficient at it. Erythro means red. What does cyte mean? Cell. So it's red cells. The epithelial cells, they all hold hands perfectly. They line up, so it's a benefit to those. It's because they form the sheet. Not just linear like that, but sideways too. So that your skin doesn't have a break in it. It will wrap all the way around your muscles, come back around, and it meets up at the beginning. It does the same thing in every direction, so it's continuous. Even at your mouth, there's no break. It just goes up, folds over, goes all the way down your GI tract, comes out your anus, and then continues with your skin again. No breaks. Some are ideal for storage, like fat cells. Fat cells can hold big wads of lipids. Here's the whole cell. That's the little nucleus, and you can see this big wad of fat. Storage for energy. Or a macrophage. A macrophage is really cool because it can rearrange its cytoskeleton so that it reaches out like these little things called their foot structures. What am I thinking? podophilia. Little feet. They stretch out, they grab a hold of the surface, they pull, stretch out another one, grab a hold of the surface and pull, and they can move relatively quick because they keep rearranging their cytoskeleton in there. And then look at that name too. Macro, what do you think that means? Yep, large, and then phage means to eat, so guess what its job is? Eating big things. Muscle cells, even within the group of muscles, you have skeletal muscles that are long and tubular, and smooth muscles that are short and kind of tapered, but they have special needs. Like smooth muscles always want to wrap around some kind of a tube, whether it's a blood vessel or GI tract or uterus or urethra or any parts of the reproductive, renal, GI tract, so that they can squeeze, like in the stomach, smooth muscles squeezing and compressing. Where skeletal muscles are designed linearly so they can pull. A little bit different. But they have the same DNA in them. I mentioned neurons before. There's the flagella on the sperm for swimming, motility.
So, smooth muscles are more like inside your cavities.
Yep, smooth muscles are all inside of you. There are some smooth muscles, like in your eye, that aren't around a tube, but most of them are around a tube. Like for your pupil, smooth muscle actually adjusts that. Still circular. Right, the next thing you have to know are when all those cells start working together to make something happen, all with a similar goal, they're called a tissue. These are the 4 types of tissues you absolutely positively have to know as fast as possible, because I'm going to keep asking you over and over again forever. Just saying. Yep, these 4. So when you get groups of these that work together for one common goal, it's called a tissue. And I like to minimize my thinking as much as possible and then work out. So right away when I think of the 4 tissues, I have 1 or 2 words that come to mind for each, and then I can broaden that out later. Like for instance, muscle tissue. What do you think its main purpose is? Move things. Yep, contraction. It's trying to move, whether it's a skeletal muscle moving your skeleton, or it's a smooth muscle propelling food or squeezing blood, or cardiac muscle that does what? Pumps blood. Yep. So they're all there to contract and move things. Nervous tissue, long-distance communication. So I always think long-distance communication from one part of the body to the next, very specifically. Epithelial, sheet-like covering. Everywhere you find epithelium, it's a protective sheet. No breaks. Like the skin or wrapping around organs. And then the last one, I always say connective last, because connective is other. If it didn't fit into a muscle contraction, nervous communication, epithelial sheet-like covering, it's connective.
Where in the body is connective tissue?
Everywhere. Yep. And we're actually going to talk about that today too. But the connective tissue is all over the place. For instance, blood is connective tissue. Just under your skin, holding your skin down is connective tissue. Fat is connective tissue. Your bones are considered connective tissue. And that's a tricky one too, because a lot of people don't think about that. A bone doesn't have long-distance communication. It's not a sheet-like covering, and it's not there for contraction. It's other. Blood's the same way. Fat's the same way. If it doesn't fit nice and neat into those first 3 categories, connective tissue. So tissues, all these different tissues working together, like the muscle, with neurons that control your muscle, and blood that flows through the muscle, and, well, muscle cells inside the muscle. They're all cells and tissues that work for one common goal, so it's an organ. And, of course, organ systems and organisms. You just keep seeing the same pattern repeating over and over and over again. So you need to know the tissue types, and as we go through them, you need to know where they're found. So like connective is everywhere. I mean, that's the simplest answer. With the nervous system, it goes pretty much everywhere. Where's the center of the nervous system? Nervous tissue? In the brain. Yeah, brain and spinal cord. So you know some locations, and of course it sprouts out from there and goes everywhere. Right. So I've said this a couple times, but tissues are just cells working together. So a tissue is a collection of cells with similar structure and function. That's the definition, or a definition. Collection of cells with a similar structure and function. Did I? Where? Yeah, thanks, spell check. Over there. And the study of tissue is known as histology. What was the study of individual cells? cytology. And then I said this before, but I would simplify things. 1 or 2 words for each of those tissues so that if you get a question, you go, well, which category would it fall into? And you don't have to overthink it and you just put it in there. So like for instance, if I'm looking at skin on this ear, what would that be? Yep, it's a sheet-like covering. How about this brain? Yeah, nervous. How about the muscle? Connective. Yep. Even though it's flat, it's not a sheet-like covering. Look at all these breaks. These sutures are actually breaks or separation areas. You've got holes throughout the skull. With typical epithelium, there aren't holes. There are no rips, there are no tears, there are no breaks. Like this. So if this were the GI tract, you can see this upper layer, the epithelium, goes all the way up, over every bump, over every groove, all the way along. It'll go all the way up through here and come down, no breaks at all. Why would you not want any breaks in your skin or the inside of your GI tract? Yeah. You separate the environment from external versus internal. You keep bacteria and viruses out. You keep blood and moisture in. You do all these things. It's a wall or a barrier between you on the inside and the rest of the world. It's protective. And then underneath that, you can see all this connective tissue holding it down. All this fuzzy, full of little tiny collagen fibers and reticular fibers and whatnot that we'll talk about too. Okay, so epithelial tissue functions. Things that I'll always start each section off with what's major functions, what's major purpose, what are the major organs that we're going to talk about, and then we'll go into fine details. So functions of epithelium: protection. It's a sheet-like protective layer, like the skin. Keeps all the bad stuff out, keeps all the good stuff that you need in. You have things like neurons that have receptors that detect your environment, so sensory. It can let things diffuse. Like in the GI tract, you have epithelium that allows passage back and forth. You have in the lungs epithelial cells that allow oxygen and CO2 to exchange or diffuse. They're also good for things like absorption and secretion. So when we talk about things like the kidneys, we'll talk about epithelial cells for filtering urine, filtering the blood, making pre-urine urine, which I just kind of said, filtration. And they can form slippery surfaces too, or they can actually create traction. Like when you're in water for a long time, the surface of your hands and the surface of your feet actually start wrinkling more so you get better traction underwater. Ain't nothing here that don't need to be here, and there's a purpose for everything.
So that's why no one else is
Right, exactly. If you've ever noticed, it's your hands and your feet that wrinkle, not your arms and stuff.
But yet people still sweat.
Because we use soap and slimy things, and people don't clean their tub, and they get all that nasty biofilm on the bottom of it. We'll get it next time. When was the last time you cleaned your tub? All the stuff, the residue you wash off you is just coating the bottom of your tub. Yep. Not very often at all. No. So, I was actually just noticing that this morning, and that's why I was thinking about it too. It's like, eh. But epithelial tissues like this, you can see the epithelium, they're cells that hold hands right next to each other. Down here, you've got all these fibers and little tiny cells all over the place. This is not epithelium. Epithelium, all the cells directly contact each other, hand to hand to hand to hand. This connective tissue is just connecting or holding down the epithelium. So when we talk about the body cavities and body surfaces, when we talk about directions, cells have direction too. Like for instance, the cell has a top, has a bottom, and it has sides. They actually have terms. Up here you have an apical region at the top. You have a basal or bottom region here, and then you have lateral sides, which you already knew that word because it means to the side. So they're all connected. When you look at the bottom part, every epithelial cell is always connected at the basal layer to something called the basement membrane. It's at the basement of the cell. It's holding or helping hold it to another tissue. So even though connective tissue is holding down the epithelium, that basement membrane is holding the epithelium to the connective tissue. It's a glue that holds glue, basically. Another important thing is it's avascular. Vascular means blood vessels. What does A at the beginning of a word mean? Without, yeah. So it doesn't have it, it's without it. So avascular means all epithelial tissue has no blood vessels, which seems weird. Yeah. So if you have a tissue that needs, an epithelial tissue that needs blood, it needs to have a blood vessel near it, but it's not in it. So for instance, your skin, you have all these cells stacked on top of each other. and you have a blood vessel running underneath it, and you have to diffuse oxygen and nutrients up to these. The ones at the base of your skin will get lots of nutrients. They'll feed and they'll replicate and they'll push new cells above and above and above. But what's going to happen to these very superficial cells that are far away from a blood vessel? Yeah, they're going to starve and they're going to die, and then they flake off.
Get kind of salty.
But epithelium is innervated, which means it has nerves. So just to show you, have you ever taken a safety pin and slid it through your skin and held it there?
Yes.
And you pull it out and it doesn't bleed? But when you do that and you poke it down just a little bit deeper and you hit one of the nerves, you can feel that damn thing. Think of a paper cut, right? You go, phew, no blood, but it hurts like a mother, and it's constantly hurting, and it's not bleeding. There are lots of neurons in there, but there's no blood vessels in there unless you go deep enough. So here you see the nerves coming up and branching into the epithelium, but the blood vessels don't. Another important thing is high regeneration, which is really important for cancers. These things regenerate quick, and you know that because you're constantly turning over new skin, sloughing off the old skin. If one of those cells becomes a cancer cell, you better hope you slough it off, because these things can replicate really, really fast, which means what about that cancer? It can spit, has potential to become extremely dangerous. One of the most dangerous is skin cancers. In fact, if you take all the other cancers that we know of, and the incidence every year of all of those, and count the total numbers, and you look at how many skin cancers there are more skin cancers every year than there are all the other cancers combined. So why don't you always hear about skin cancer killing people constantly?
Because it's easy to get rid of.
Yeah, we're vain people. We can see when something's ugly growing on our skin. We go, ooh, cut it off, right? And it's gone. It's really easy to treat. It's really easy to identify. Where if you have something growing inside of you, you have no clue until it's big enough that it's causing problems.
Did you hear that?
That sensor is right up there, and I was hoping something moving by it would trigger it. All right, and then they have specialized connections between them that hold them together, and we're going to look at these. So you can see all these special connectors that hold one epithelial cell to the next. Here are the three types that you have to know. So look at them: tight junctions, desmosomes, and gap junctions. You can see examples of them down here, and I'll just blow these up for you. So there they are. There's the first one. So tight junctions, I always think of them being like Velcro. Is Velcro strong and unrippable? No. So you've got one side of the Velcro on one cell, the other side on the other, and if you pulled them hard enough, you'd actually just peel them apart. The benefit to tight junctions is that they're waterproof. They seal the cells really well. But unfortunately, they're not very strong. When you see them, they're up towards the surface of the cell. So if this is the GI tract and you see all the bumps where the food would go, you'd see them right there so that particles can't pass in between, or water can't pass in between. It's a barrier. The second one's the ones that are strong, are called desmosomes. My family used to do concrete work, and when they would put together driveways, I always thought it was interesting. They'd throw what's called rebar, or some people call it rewire. They'd throw all these metal wires inside the driveway and then dump concrete over it. And the reason is because concrete, it can handle a lot of weight, but if you shift on the weight just a little bit, you crack it. So that's not good. The wire, you can't just build your whole driveway out of wire, because wire bends way too easy, and it doesn't give a lot of support. So what they do is they put the wiring in, and then they pour concrete over it, so the two things hold each other together really well. It's the same thing with the desmosome. You see all these protein wires that go back and forth, weaving, like stitching, holding the two cells together. And then it pours a calcified plaque over the top of it to kind of glue it down and make sure it's not going anywhere. It's really, really strong. So anywhere that you get a lot of tugs or pulls, you're going to find a lot of this desmosome, these desmosomes, like in the heart. 24 hours a day, every second your heart beats, hopefully. Constantly tugging. You don't want those cells pulling apart. And the last one, gap junctions, and they're just little gaps. They're an open pore or open hole in between two cells. So if this cell needs to communicate with the next one, it can send something like calcium through. And that calcium can be a chemical messenger. They're not there for strength at all. They're there just to connect one cell to another so they can talk to each other, communicate. So like these smooth muscles, if my GI tract is an example, if I want to squeeze the whole stomach, But one cell fired at a time, it would be so difficult to do that. Instead, I fire one set of cells, and they communicate to all the other ones around that chain, so they all fire together as one unit, squeeze. Gap junctions. And in this picture, I borrowed from another textbook, but it shows all of them together and gives a little bit more detail, if you want more detail.
So,
Yep, like smooth muscle. So a lot of smooth muscles are connected by these gap junctions so they can work together. In fact, the tissues in your heart, the cardiac cells, they're connected by gap junctions too, so that one chamber of your heart, when part of it contracts, the whole chamber will actually contract in unison. Next important feature, epithelial tissues can actually make glands. Glands are made from epithelium. They're just sheets that bend or bow in. And there are two types of glands, specialized glands. One's called an exocrine and one's called an endocrine. Guess which one pushes stuff out of the body? Exocrine. Yeah, so things like sweat glands, oil glands, ceruminous glands, which make earwax, those are all exocrine glands.
Earwax get there. What's the problem?
There are glands inside your ear, so it makes it, it's supposed to be an insect repellent, is what it is.
Oh, I put a bug in my ear. It is really bad.
The bug probably got lost and took a wrong turn. There was an article one time that I read about, and it showed this spider in a woman's ear. She was having really bad problems, and the doctor went and looked in, and he took a picture of it, and there's just a spider peeking out over the...
I saw, I think there was a video like that I saw.
I like that curl around the corner and curl back in.
It is gross.
My gosh. Wake me out. Wake me out.
Hold on,
Like I feel like snake and flies in my ear, like flying around my ear, and I was like,
Yeah, I hate it when any kind of a bug flies by my ear. It drives me crazy.
and my dad wouldn't let me go to the ER. He made me come to work the next day, maybe something in my ear, and like tried to like get out with like a paper clip. I was like, you suck so bad. It hurt. We got it out, but it hurt.
But those are all examples of exo. They're pushing to the surface of the skin. You know, inside the ear is still in the surface of the skin, sweating, oils releasing. And then endocrine pushes towards the blood. Endocrine always pushes out something called a hormone, and it's trying to push it into the blood as a chemical messenger. Endo and exocrine. The basement membrane is another important feature. Like I said, with the epithelium that goes all the way along, it has to be glued down somehow. So the glue is the basement membrane, and it holds it to what other tissue? Tissue. So remember, there are only 4 types of tissue. Epithelium is held down by the basement membrane to what? Connective tissue. Yeah. And you can kind of see that nice dark line right along the bottom here. So these are all epithelial cells. I believe they're in the trachea by the looks of them. Does it say down here? Or doesn't it? So all these little things are called cilia, so that if you inhale bacteria or something, you can grab a hold of it, and the cilia will actually move it along so you can push it up in your nose to pick it later, or you push it in your throat to swallow it and eat the bacteria. One of the 2 pathways. And then here you see all these cells holding hands. They're tightly packed. It's definitely epithelium. And right along the bottom, you can see that dark basement membrane that glues it down to all this connective tissue.
So like that's pseudo-stratified colon or whatever. Why is it without versus the other?
Did we already talk about this, or did you bring it up? Okay. Let's say we're supposed to talk about it today.
Why the nuclei different?
That's a great question. We'll answer in just a little bit. Okay. So, nice segue. When you identify or you label the tissues, there are 2 ways that you label them when they're epithelium. And I should have switched this around because technically the first thing you do is you say how many layers it is. If it's 1 layer, they call it simple. If it's 2 or more, doesn't matter if it's 1,000, it's called stratified. So when you're writing the name or identifying something, you always start with a number. Is it simple or is it stratified? Then you look at the shape. There are 3 possible shapes. Either it's squashed, we call squamous, or it's a cube, which we call cuboidal, or it's columnar, which looks like columns. It's that easy. So if you see a layer of cells that, what's the first thing you have to call it? First.
Stratified.
Yep. So why stratified?
There's 2 layers.
Yep, they're 2 layers. So, and by the way, here's the fuzzy basement membrane on the bottom, so we know where top and bottom's at. 2 layers, so it's definitely stratified. And then identify the shape. Cuboidal. Stratified cuboidal. So simple squamous, you can see it's 1 layer. Just look at the nuclei. 1 layer thick, squashed. 1 layer thick, cubed. 1 layer thick, columnar. Usually with the columnar, the nucleus is closer to the basement membrane though. Not always, but usually. And when you look at simple squamous, or any squamous in general, even the nucleus is squashed. So those are all simple. When you're looking at many layers, the trick to labeling the many layers, because look, at the top of this it's squashed, but at the bottom it's what? Looks cubed. You name it by the top or the apical surface. Whatever's closest to the surface is what you call it. So even though it starts as cubed at the bottom, by the time it's at the top, it takes on its true shape, which is squamous. And since there are lots of layers, it's stratified. And there's stratified cuboidal. And the pseudo-stratified, pseudo means false. So it's not really more than 1 layer, it's 1 layer. If you look at this, the trick is that every one of these cells in this touch the basement membrane. They're all technically 1 layer across the basement membrane. But some of them are tall, fat at the top, narrow at the bottom, so their nucleus gets pushed up. Others are short, where the nucleus is down low. So if you look at it, it looks like a row of nuclei here and a row of nuclei here, which makes it look like more than 1 layer. But the trick is that every one of these cells touches the basement membrane, every one of them. And then transitional. So transitional can change its shape. If you pull on it, like if you're talking about the bladder, it can change from being a cuboidal to a squamous, or a columnar to a cuboidal. It can transition. So they call it transitional. On microscope. Yeah. Not all of them, but a lot of them. So when we do start identifying them, you need to name them, tell me how many layers, what their shape is, and then tell me where they're at and a main purpose. So for instance, when you see stratified cuboidal, typically you find it lining ducts and sweat glands. It's good for secreting things like ions and water. When you look at something like, well, simple squamous, it's thin and it's perfect for diffusion where you want transport rapid. So it's going to be in places like the lungs or the blood vessels where you're trying to exchange oxygen, CO2, nutrients, electrolytes. Uh-oh. I knew it. It froze. Okay, maybe that's just a way of saying it's a good time to take a break.